Welcome to Methodical Medics, and our second video on cellular respiration. This time we're covering the transition reaction, which bridges glycolysis and the TCA cycle. It's a short but crucial reaction, and I've made two animations for high schoolers and for MCAT or university students. At the high school level, it's a very simple three-step reaction. Remember that glycolysis was done in the cytoplasm of the cell and needed 10 enzymes. Well, the bridge reaction occurs in the matrix of the mitochondria, and it's entirely catalyzed by the pyruvate dehydrogenase complex. In step 1, pyruvate loses a carbon dioxide to form a 2-carbon molecule called an acetyl group. In step 2, another molecule called coenzyme A covalently bonds to the acetyl group and forms acetyl-CoA. Coenzyme A acts like a carrier for the acetyl group, so it can properly be harnessed in the TCA cycle. In the third step, NAD gets reduced to NADH and a proton, and it moves on to the electron transport chain. This equation below summarizes the bridge reaction and its three steps. Our reactants were pyruvate, NAD, coenzyme A, and were converted into acetyl-CoA, carbon dioxide, NADH, and a proton. See, nice and simple. This is all you need to know at the high school level, but if you're studying for the MCAT or university biochemistry, then the next animations are for an in-depth look. Now we're going to look at the bridge reaction in higher detail, starting with how it actually enters the mitochondria from the cytoplasm. First, pyruvate must go through the outer membrane. It does this by facilitated diffusion through a voltage-dependent anion channel, or VDAC. Next, pyruvate must pass through the more selective inner membrane. This is done by secondary active transport through a mitochondrial pyruvate carrier, or MPC. MPCs are symporters that use a proton electrochemical gradient to drive pyruvate and a proton into the matrix, where it is ready to be processed by the pyruvate dehydrogenase complex. This is single-handedly the most important animation for MCAT takers, showing the in-depth version of how pyruvate gets converted into acetyl-CoA. The only step up from this would be the reaction mechanism, which isn't really necessary. Earlier I said that the bridge reaction was catalyzed by PDC, but if we actually zoom in, it's composed of three enzymes working in tandem. Imagine the PDC like a factory assembly line, running 3 cycles at once, where pyruvate is like an unpolished rock that needs to be processed. It goes into the first compartment, E1, pyruvate dehydrogenase. Within this enzyme is a prosthetic group called TPP, thiamine pyrophosphate, which is derived from vitamin B6. TPP acts like a factory arm, which holds and slices an unwanted piece from the unprocessed stone. That part being trimmed is a carbon dioxide. So E1 decarboxylates pyruvates, and now we're left with an intermediate called hydroxyethyl TPP that is ready to move on to the next step. In E2, dihydrolipoyl transacetylase is the second compartment, and lipoic acid is another prosthetic group that acts like the next arm in the assembly line. It has a disulfide bond in its oxidized form, which acts like a glue and polisher for the rock. The disulfide bonds take the hydroxyethyl group from TPP and oxidizes it into an acetyl group. TPP is reset for another pyruvate, which completes the cycle in E1. Next, coenzyme A is like a handle, which makes the rock easier to process in the TCA cycle. The sulfhydryl group in coenzyme A bonds to the acetyl group and replaces lipoic acid, forming acetyl-CoA. However, we're left with dihydrolipoyl, the reduced form of lipoic acid, so this process is not quite finished yet. Finally, E3, dihydrolipoyl dehydrogenase, functions as the last compartment. It uses FAD to oxidize dihydrolipoyl back to lipoic acid. This is like re-adding glue to the arm, and the second cycle is complete. It is ready to accept another group from TPP. FADH2 donates its electrons and hydrogen to NAD, forming NADH and a proton, which finishes the third cycle. So why not just reduce NAD right away into NADH? That's because FAD is a prosthetic group which is tightly bound to E3, while NAD can freely diffuse to function in other parts of the cell. So the cycles are complete and can repeat with another pyruvate. So to summarize, we start with 1-glucose, which turned into 2 pyruvates after glycolysis. The bridge reaction turned them into acetyl-CoA, CO2, NADH, and hydrogen, all in multiples of 2. Thanks for tuning in. I hope this video was a drastic improvement from glycolysis, as I've learned new animating skills, and I'll see you in the TCA cycle.